Transcription is essentially the process in which we go from DNA to RNA. There are a few different things that happen to get us from DNA to RNA, and there are also a few different types of RNA, including tRNA, mRNA, and rRNA. The main objective of this video will be focusing on the transcription of DNA to mRNA. Eukaryotic transcription is carried out in the nucleus of the cell and proceeds in three sequential stages: initiation, elongation, and termination.
Unlike the prokaryotic RNA polymerase that can bind to a DNA template on its own, eukaryotes require several other proteins called transcription factors to bind to the promoter region and then help recruit the appropriate polymerase. In eukaryotes, transcription is carried out in the nucleus of the cell. DNA is transcribed into pre-mRNA, which then goes on to processing and then to translation into a protein. So let's see how this works.
The primary actor in the process of transcription is RNA polymerase. RNA polymerase II will be used to create a nucleotide sequence if we are coding for mRNA. If we were coding for the transcription of tRNA, then RNA polymerase III would be our main actor. And if we were coding for the transcription of rRNA, RNA polymerase I would be the main actor. What this tells us is that the type of RNA polymerase that binds to the DNA all depends on what we are coding for. To kick the initiation process off, RNA polymerase needs to know where to start. It begins by attaching to a promoter. Every gene, especially in eukaryotic cells, will have a promoter associated with it.
Within this promoter is what is called a data box, and is essentially a DNA sequence that indicates where a genetic sequence can be read and decoded. A core promoter is that portion of the proximal promoter that contains the transcription start sites. The data box, as a core promoter element, is the binding site for a transcription factor known as data binding protein, which is itself a subunit of another transcription factor 2D. Transcription factor 2B binds to transcription factor 2D and then enables RNA polymerase II to bind to the core promoter. After transcription factor 2D binds to the data box via the data binding protein, 5 more general transcription factors, including transcription factors 2F and 2E, which have not already been mentioned, and RNA polymerase combine around the data box in a series of stages to form a pre-initiation complex. One transcription factor 2H, is involved in separating opposing strands of double-stranded DNA to provide the RNA polymerase access to a single-stranded DNA template. Once the RNA polymerase attaches to the DNA strand, it can then separate those strands. However, only a low or basal rate of transcription is driven by the pre-initiation complex alone. After the DNA is separated into two single strands, elongation occurs. The RNA polymerase can code for the RNA in the 5' to 3' direction. However, there are other actors that are involved with the promoter. These are called transcription factors, and these factors control the regulation of the transcription process. Other proteins known as activators and repressors, along with any associated co-activators or co-repressors, are responsible for modulating the transcription rate. Activators help bind the DNA polymerase to the promoter and can increase the rate of transcription. Co-activators activate transcription from a RNA polymerase II promoter. It does not bind DNA itself. Activator proteins increase the transcription rate, whereas repressor proteins decrease the transcription rate. Transcriptional repression in eukaryotes is achieved through silencers. The two types of silencers are silencer elements and negative regulatory elements. Silencer elements are classical position-independent elements that direct an active repression mechanism, and negative regulatory elements are position-dependent elements that direct a passive repression mechanism. It is the silencer itself and its context within a given promoter rather than the interacting repressor that determines the mechanism of repression. In some cases, eukaryotic repressors simply interfere with the binding of other transcription factors to DNA, such as RNA polymerase. Silencers form an intrinsic part of many eukaryotic promoters, and consequently, knowledge of their interactive role with enhancers and other transcriptional elements is essential for our understanding of gene regulation in eukaryotes. The side of DNA that RNA polymerase interacts with is called the template strand. If you think about the information that the RNA will encode, it will actually contain the same information as the coding strand of DNA. The only exception to this is that you will not find the nucleobase, thymine, and RNA. You will instead have uracil. So after all of this is set up, the transcription factors, the RNA polymerase, and a mediator protein that serves as the interface between the RNA polymerase and regulatory transcription factors, signals transcription that will commence in the 5' to 3' direction until the 3' end of the transcript is cleaved near the poly A tail signal sequence. The termination of transcription leads right into the processing stage of mRNA. External stimuli such as methamphetamines are introduced to the brain. The CAMP responsive element binding protein, or CREB, begins to be expressed at high levels to help the brain adapt to its environment. ICER, or inducible CAMP early repressor, is a way to stop the stimulus by methamphetamines. Let's see how ICER works. This image is a mechanism of transcriptional repression by AZIP proteins. These mutants retain the leucine zipper required for dimerization. The basic DNA binding domain, however, is exchanged for a domain containing acidic residues. Dimerization between a wild-type B-zip protein with an A-zip repressor molecule inhibits DNA binding of the wild-type B-zip proteins due to the blockage of the DNA binding domain. When ICER comes in as a response to the high levels of CREB, it overexpresses itself and can abolish the meth-induced increase of CREB expression.